All right, so this time we're going to be talking about Chapter 5, Lesson 3, and so we're going to be talking about DNA. DNA stands for deoxyribonucleic acid. So DNA, deoxyribonucleic acid. And what that is an organism's genetic material. So all of that stuff that we've been talking about with the chromosomes and the genetics and the heredity and all of that. So all of that genetic material, the stuff that makes you, you, is all contained on that DNA. So DNA, remember from the chapter 3 and 4 where we talked about mitosis and meiosis, it coils up to make chromosomes. And we mentioned also last chapter that a gene is a segment of DNA on a chromosome. And you can see that right here if you look at the picture. DNA also contains the directions an organism needs to grow, to maintain itself, and to reproduce. So all of that stuff is contained on the DNA. So let's talk a little bit about how DNA was discovered. In 1869, there was this guy, and I can't really say his name, Frederick Miescher, maybe? And he was the very first person, though, to identify DNA. So he's the very first person who saw this stuff and went, wonder what that is. Must be something special. So he identified the DNA first. Then in 1919, Phoebus Levene published a paper about nucleic acids. Now remember, DNA is deoxyribonucleic acid, so DNA is a nucleic acid. In 1950, Erwin Chargaff came up with rules for how DNA is put together. So what Chargaff said is that the number of adenine in DNA always equals the number of thymine. So A equals T. And the number of cytosine in DNA always equals the number of guanine. So C equals G. Chargaff's rules became very, very important to the next people. And you can see here there was a pretty big gap between Phoebus Levene talking about nucleic acids before 1950 when Chargaff actually came up with his rules. So those 50s is when this whole idea of let's figure out what DNA looks like really, really, really took off, I guess. So in 1951, Rosalind Franklin and Maurice Wilkins used X-ray diffraction to get images of DNA. Rosalind Franklin was pretty amazing that she was one of these people because she was a woman, and in the 1950s, they didn't really think that women, should be scientists and whatever. So for her to be a scientist and to be able to have this X-ray diffraction images of DNA and to really have contributed to the finding of the shape of DNA is pretty cool. Unfortunately, Rosalind Franklin, Maurice Wilkins, and then the two people who actually finally decided what DNA looked like, they didn't always all get along. And perhaps, if Watson and Crick, who finally discovered the shape of DNA, if they had actually, looked at Rosalind Franklin's results and actually taken her a little more seriously, they probably would have come up with their idea a little bit sooner. So her X-ray diffraction images showed that DNA has that spiral shape. And that's going to be important. So then in 1952, Alfred Hershey and Martha Chase, again another woman who contributed to this, did some experiments with viruses to show that DNA actually carried genetic information. So that's a pretty cool thing too. Then finally in 1953, we have Watson and Crick's model of DNA. So James Watson and Francis Crick finally built a model of DNA. And their model of DNA showed that DNA had what we call a double helix shape or a twisted ladder, or some people say it looks like a spiral staircase. So they are the ones who really came up with the model and the idea of what DNA really truly looked like, so how it was put together. Okay, so DNA has a double helix shape, so, or that twisted ladder, whatever you want to call it. DNA is pretty specific in how it's laid out. So it's made up of these things called nucleotides. That's what makes it a nucleic acid. So nucleotides are made up of a nitrogen-containing base, a sugar, and a phosphate. And if you look at the picture here, you can see the nitrogen-containing base, the sugar, the phosphate. So one set of those is called a nucleotide. The sugar phosphate groups are going to form the sides of the DNA. So like the sides of the ladder. The nitrogen bases are the rungs of the ladder, so the steps on the ladder. And there are 4 of those nitrogen bases. So the first one is adenine. Then we have thymine. Then we have guanine. And then we have cytosine. So A, T, G, C. Those are the 4 nitrogen bases that are involved in DNA. When those bases pair up, they're going to pair up in a really specific way. And we say that they're complementary. So if you think back to Chargaff's rules again, he said there's the same number of adenine as thymine. So what Watson and Crick discovered was that adenine and thymine always bond together. Same thing with cytosine and guanine. So A and T bond together, C and G bond together. The order of those nucleotides, so the order of those bases really, is the code that carries the genetic information. So that's what decides what you look like, really. So it's the code. It's kind of cool, I guess. Okay, the next 3 things we're going to talk about are going to be a little more confusing. And we're going to start here with DNA replication. So what does replication mean? Replication is copying, right? So you make a replica, you make a copy. So DNA replication is the copying of the DNA molecule to make another identical one. And you can see in the picture, you can see what's happening in the process of DNA replication. This is going to happen before cell division. Remember back when we talked about mitosis and meiosis, we talked about how during that interphase, the cell was getting ready to divide, and so you had the DNA copying itself and all of that kind of stuff going on. This is what they were talking about. This is what we were learning about, this DNA replication. Okay, so the first thing that happens is that the DNA molecule is going to split in the middle where the bases meet. So it kind of separates, and you can see that in the picture there. Then complementary nucleotides are going to be added to each side. So remember, A is going to pair up with T, T is going to pair up with A, C is with G, G is with C. So bases on each side are used as a pattern for the new DNA. So if you had like an ACG on the one side, then the new bases would be a TCG. So then finally what happens after you get these complementary nucleotides added, you're going to end up with 2 new identical DNA molecules formed. So each of those are going to be made of half of the old DNA and half of the new DNA. So you've got a copy. The next process we're going to talk about is going to add another nucleic acid for you guys to know about, and that's going to be RNA. So RNA is ribonucleic acid. And RNA is a little bit different than DNA. First of all, it's got a different sugar. And it's also just a single strand rather than a double strand. And then one other thing that's different about RNA from DNA is that RNA does not have thymine. Instead, it has something called uracil. So there's no T, T is replaced with a U when we're talking about RNA. So RNA, ribonucleic acid. RNA's job is really to help make proteins. And what it can do to help us make those proteins is it can create a temporary copy of the DNA, which is really important in the building of proteins. So again, RNA is made up of a single strand, so it's not a double helix, it's a single helix. And it also has that sugar phosphate backbone, just like DNA does, except that it's a different sugar. And then it still has those nitrogen containing bases. And like I said, you still have adenine, you have cytosine, you have guanine, but there is no thymine, instead you have uracil. There are 3 types of RNA. We have messenger RNA, ribosomal RNA, and transfer RNA. So messenger RNA is going to be the temporary DNA copy. The ribosomal RNA is part of a ribosome, and it's going to help read that temporary copy. And then the transfer RNA is going to be the stuff that helps bring that the protein building stuff to the ribosome to help build that protein. But we'll talk about all of that a little bit more in depth when we talk about the protein synthesis and translation part of the stuff we're going to be learning about. Okay, so the next process, so we had replication, so that was copying the DNA, making a copy of the DNA. This time we're going to talk about transcription. So what transcription is taking the DNA information and copying it to the messenger RNA. So this is that temporary copy of DNA that we're talking about. So it only happens with individual genes. So it's not going to be an entire strand of DNA that's going to be copied into RNA, mRNA. You're only going to have little individual genes and instead of the whole molecule. And you can see that in the picture. So the steps to transcription are as follows. The first thing that happens is the DNA is going to open up at a gene. So it's going to split apart at that gene. Then you're going to have RNA bases matched up to the complementary bases on the DNA template. So if your DNA was an A, T, C, G, then your RNA would be a U because U would match up with A, then an A because A would match up with T, then a G because G matches up with C, and then a C because C matches up with G. So it's still that same complementary idea that we used when we were talking about the replication, except that we're using RNA bases this time instead of DNA bases. Okay, when that process is finished, then the messenger RNA gets released and the DNA molecule closes itself up. So that's transcription. So that's copying that DNA information to the messenger RNA. Okay, so how does DNA work? Basically what we have with DNA is that it's holding that genetic information, so that code, in groups of 3 bases. And we call those groups of 3 bases a codon. The codons are actually the instructions for making proteins. Proteins make up basically all of your body parts and how they get put together is all dependent on this whole codon business. So you've got your 3 bases, or your codon. So each group of 3 bases, so each codon codes for a specific amino acid. And those amino acids get put together to form proteins. So that's kind of the whole point of DNA. So how do we make those proteins? We're going to use this process called translation. So what happens is the information on the messenger RNA gets used to build the protein. And you can see in the picture how that works. So the first thing that happens is the messenger RNA goes to a ribosome. Remember, ribosomes are made of rRNA, so ribosomal RNA and protein. The ribosome is going to read the base sequence 3 at a time. So it's going to look at those bases 3 at a time and read that. So it looks at the codon. Remember, a codon is 3 bases. So the ribosome reads the codon. And it's basically going to sort of tell the transfer RNA to go pick up a specific amino acid based on what the messenger RNA code says. So the transfer RNA goes out, picks up the specific amino acid that it's supposed to, and brings it back to the ribosome. And then this process continues. So more transfer RNAs go out and pick up more amino acids and bring them back. And those amino acids get lined up and linked together to form proteins. And then when you hit the end, then it gets released and you have this lovely little protein that does whatever it's supposed to do. Now, these processes sound, pretty simple and straightforward and easy when we talk about them here. Obviously, we've simplified them way down for you guys because you're just 7th graders. But sometimes we make mistakes. And so when there's a mistake that gets made, that's called a mutation. So mutations can be changes in the number or the type or the order of the bases on the DNA. There's a lot of different things that can cause mutations to happen. They can be triggered by exposure to X-rays or UV light or radioactive materials or chemicals or lots of other things. And there are 3 types of mutations. So remember, this is changes in DNA. That's what a mutation is, a change in the DNA. So there's 3 types of mutations. The first one is called deletion. In deletion, what happens is one of the bases is left out. So and most of this is happening during that replication phase. So you've got a base that's left out, so a whole base missing. The second kind is called insertion. If you have an insertion mutation, you have an extra base added. And then finally, the third one is substitution, which is just using the wrong base. And this kind is the most common. Mutations are actually more common than we probably think they are because, if you think about it, you've got all of these cells, these millions and millions and millions of cells in your body, right? And they are all going through these processes and replicating and making new cells all of the time, right? So this whole replication process is happening millions and millions and millions of times. Well, you can't expect it to be perfect. Nothing's ever always going to be perfect. So quite often you have some sort of whoopsie that happens. So you end up with a mutation. So there are some consequences of mutations. The first one is sometimes it improves the trait. Sometimes it makes the trait better. And we'll talk more about this kind of thing when we get to the chapter on natural selection and evolution and that kind of thing. And we'll talk about how improved traits happen. And so you end up with these better traits through mutations. Sometimes, well, most of the time, there's just no change. Nothing happens. There's a mutation, it doesn't affect anything, and everything's just fine and dandy. And then the third thing that can happen is you can have a harmful trait. So what happens with the harmful trait is that the trait actually is worse than it normally would be. A lot of those harmful traits lead to what we call genetic disorders. So genetic disorders are like, we can kind of think of them like diseases related to genetics that are caused by these harmful mutations. Genetic disorders can be inherited mutations. So it can be a mutation that's passed from parent to offspring. Examples of that would be like Tay-Sachs or sickle cell anemia or Huntington's disease. Or you can have a mutation that's developed during one's lifetime. And an example of that would be like cancer. All right, so this ends Chapter 5, Lesson 3.